I'm really happy to be here and share some of the work we've been doing with support from CFRI and others. And today I thought I would just cover and it'll seem really basic. I realize I just want to bring us all to the same page. But I'll cover, antimicrobial resistance. Sorry. And bacteriophage therapy and bacteriophages and all that. And then I'll go into some of the research we're doing and go over a brief summary of some of the cases we've treated, CF related. So AMR infections, you guys are, I'm sure, all very, very familiar with them. Um, but how do they evolve? I'm sure maybe you're familiar, but it's actually one of the critical parts to the research we do here. Um, and so typically we treat bacterial infections with antimicrobials, as you know, um, and they work more like, they just, it's a bomb basically that goes off and kills a bunch of bacteria. However, it doesn't kill 100% of them all the time, and any survivors will go on to grow. You'll get a recurrence of symptoms, and maybe you'll use antibiotics again. Except this time, resistance has evolved. So, and I'll try to be quick in my talk here so we can regain some of the time. I keep my eye on the clock here, so I'll try to be done in 40 minutes or less. Anyway, once this, when you have bacteria that don't respond to antimicrobials, you have antimicrobial resistant or superbug infection. And it's as I'm sure you all know, it's a major threat to human health. And by in just 30 or so years, 10 million people are going to die every year from AMR. And there are actually bacteria now, as I'm sure you all are very well aware, that are resistant to all available antimicrobials. And individuals that have CF are at the edge of this crisis, as you know, the amount of AMR infections is really high. Um, but how does it work, right? So I know we're all familiar with AMR, and I apologize for the cartoons, but I couldn't find any ready-made graphics, and I'm not the best, but I tried. So there are tons of different ways that antimicrobial resistance can occur, right? So, and this is just a quick summary of a few of the mechanisms, but many bacteria, especially those causing pulmonary infections associated with CF, have redundant mechanisms, right? So they'll have many of these mechanisms existing in one population. So there's enzymes, right? And this is so before the antimicrobial can even get in the cell, there's some sort of enzyme that just breaks down or inactivates the antimicrobial and prevents it from functioning. There's antimicrobial modification where there's something produced by the bacteria that breaks the antimicrobial or modifies it. There's target alteration. So each antimicrobial has a really specific mechanism of action. And so maybe that's binding to a particular protein or sugar or something. Well, in some bacteria that site is modified, which prevents it antibiotic from doing what it needs to do. Okay, and there's antimicrobial efflux, and this is where the antibiotic actually gets into the bacteria, and but before it can cause its effect, the bacteria will just pump it right back out. So it'll keep doing that until, and normally you just hit it with more antibiotics, but then it'll just increase the number of efflux pumps and keep pumping it out. Antimicrobial impermeability is where the antimicrobial just can't get into the bacteria because of a cell wall or other modification. It just can't get in. There's horizontal gene transfer, which is particularly scary because this is where one bacteria that has resistance genes can transfer them to another bacteria, oftentimes not even the same species, and sort of transfer resistance via one of these other styles of antimicrobial resistance. Um, and so that's like, a big problem. And there are other, mechanisms that um, have been discovered that I just didn't explain, and many more that will be discovered. Um, uh, but that's just some of them, right? And so, since no real chemical antibiotics are being developed for a bunch of reasons, whether that's economic or other reasons, people have started looking into non-chemical alternatives, nanoparticles or working antibodies or other bacteria, bacteriocins. But today we'll talk about bacteriophage, um, which are these guys. So this is sort of your stereotype bacteriophage. They're also called phage, and they're just the viruses of bacteria. Um, and they basically look like this, but here are some actual pictures of them. EM images on the left here. And they have all sorts of different shapes and styles. And that's the three major groups on the old classification, but there are many, many more now. But they're the most numerous organism on the planet, and so estimates of like 10 to the 31st. Um, and they were discovered about 100 years ago. And in the lower right here, there's a QR code for a book, just a Google search of a book that was written by a medical historian who's a really great guy, about Felix Durrell, who's the guy that discovered bacteriophages in like 1914-ish. But um, they were discovered before antibiotics, chemical antibiotics, really, and so they're immediately started being considered for use. However, uh, for a bunch of complicated political and economic reasons, that just never really were used in the U.S. at least, and Europe until the antimicrobial resistance crisis started getting worse and worse. And so recently they've been, they goes through waves of interest, but then recently they've been getting a lot more attention as one of the potential solutions for for AMR infections. So we find phages anywhere we can find bacteria. They're like everywhere. So we spend a lot of time searching through sewage, actually, because that's a great place to find human-associated bacteria. And so where the bacteria are, that's where you'll find the phages. Anyway, so so the life cycle, there's more than one, but for the simplified purposes here, we'll just illustrate one sample. And I apologize if I'm all scattered. It's definitely COVID brain season. But you start off with phages here, free floating in the environment. And then they bounce around and they come in contact with one of these bacteria. They bind to it, inject DNA or RNA, and then they produce new phage particles, assemble them, and then they burst, right? So in a simple closed system, like in a flask or something, you'll have this cycle repeat and amplify more and more phages. However, just like they've evolved resistance to chemical antibiotics, bacteria can evolve resistance to phage as well. And so the work that we perform at the lab is we sort of anticipate this and try to exploit it therapeutically, creating trade-offs, which is where on one side, the left here, the bacteria is sensitive to phage, but is virulent or antibiotic resistant or something we don't want. And then on the right is after exposure to phage and resistance evolves, the bacteria are phage resistant, unfortunately. However, they've lost their virulence or ability to be antibiotic resistant or some other clinically important factor we're able to capitalize upon. So in our minds, like therapeutically, it would be a beneficial thing when the phage kill the bacteria, of course. But if they evolve resistance, we're hoping that we can also exploit a trade-off and that the therapy is also at least successful because we'll have more tools to manage the bacteria. Okay, so if you remember the first step of that life cycle, it involved binding, the phage binding to the bacteria. Now, phages are incredibly host specific, and it's actually quite a complicated process that's like 10 pay grades above my understanding, but it's really complicated, requiring the phage tail fibers, the little legs at the bottom of this moonlander looking guy, um, to a specific receptor or receptors sometimes, right? So as it's bouncing around in the environment, um, it'll grab onto stuff, and if it's not the right one, it'll fall back off again, right? And these proteins or sugars or whatever are not present on all members of a genus, and not even all of a species, right? So you'll have a phage that's specific to one, occasionally specific to one strain of Pseudomonas aeruginosa, right? And so it can be very, very, very specific. And then others, we have, staph phage that can infect and kill, most strains of Staph aureus that we see, at least clinical strains. So, many phage are hyper-specific, and others what we'd call broad host range or less specific, but it's nowhere near the sort of host range of, or not host range, the killing spectrum of like chemical antibiotics. Say. So, however, once it finds its appropriate receptor, then the bacteriophage binds and then begins the life cycle. So this super specific binding is what we really focus on in the lab. So we try to find phages that bind what are called virulence factors, or proteins involved in antibiotic resistance as well, which some would consider a virulence factor as well. For example, try to move this out of the way. Okay. For example, consider the type 4 pilus, right? So my cartoon of the bacteria on the left with all these hair things is supposed to be a representation of a pilus, and here's an image that I took from the internet. It's actually from this paper. It's a really old one. I think it's probably older than most or all of us, in the room or in virtual here. But I feel like the old EM pictures, I think they just did better work on EM. I've seen quite a few sloppy images. Definitely have produced quite a few myself. There was like an art to it that I just don't have the skills for. Anyway, you'll see these little hair-like things at the end of this, the black blob here at the, I guess I'm pointing at something, it doesn't really help, but the black blob in the lower left of that image is the bacteria, and then those little hair-like things coming out are the type 4 pilus. And that's involved in like attachment to the epithelial cells, or it's also involved in twitching motility. So it's how it like twitches around and moves. There's other ways to move as well, which are also targets. But in this case, we're just looking at type 4 pilus. Okay, so we have phage actually that require this pilus as their receptor binding site. So they can grab onto these, and that's how they infect the bacteria. And when the bacteria evolve resistance, they drop the pilus, and they're no longer able to do this twitching motility. And there's a cool paper here in this other QR code by Sylvia Worsley, who was visiting our lab from Germany a couple years ago now, who did some really cool work where she exposed the lab strain of Pseudomonas aeruginosa PA01. She studied that one. Here's the control here on the left. And then she had a knockout of one of these pilus genes, which is this delta PIL-Q next to it. And what she did was she measured the area here is how far the bacteria move. So it's like a special assay where they test the ability of the bacteria to twitch and move through some agar. And so when you knock the pilus out, it can't twitch, so it doesn't move. That was for control. And on the right side, she had a strain of Pseudomonas that was sensitive to this phage. And then she evolved resistant mutants, and then she tested their motility, and those guys are on the right there. And then you can see that there was a big change in ability of the resistant mutants to move, right? And so it was basically creating a knockout via phage. So those phage resistant guys lost their ability to, or they lost their pilus and therefore their twitching motility. And some other work later, we have other papers that are, I should have put here, but that show that these mutants are unable to, adhere to the airway epithelium. And that's some really cool work done by Gail Stanley, who's a pulmonary physician at Yale. And if you email me at the end, I'll send you her paper. I really should have put it here. Sorry, Gail, if you're watching. Okay, so another example would be like the capsule. So in some bugs like Klebsiella, there's a capsule that prevents it from being identified by the immune system. And we have phage that actually require this, and then the resistant strains don't have a capsule and have attenuated virulence. And then infectious disease fellow Shiv Gandhi is producing a paper, and hopefully that'll come out soon. If you email me, I can get it to you once it comes out, or just keep your eyes open. But there's a bunch of other targets that we have phage for in our library, right? So we have phage that can prevent gene exchange by targeting a pilus, a conjugative pilus. We have phage that can reduce or prevent exotoxin production or quorum sensing. We have phages that can either, in the resistant mutants of bacteria, can no longer form biofilm, or we also have phages that can cut through biofilm to infect bacteria that are in the biofilm. And so if something's a virulence factor, we're probably chasing it down, trying to find phage that can force a trade-off, whether it's in Pseudomonas or Klebsiella, E. coli. We're looking at all the escape pathogens with hopes that we can develop a big library of phages that can drive these trade-offs. Okay, so then you have all these phage that can, drive these trade-offs. So then the next question we get asked a lot is whether we should just throw all these phage into one cocktail, or whether we should use them sequentially, right? And there's a big, I guess, it seems like a difference of opinion in the field, whether we should go one way or the other. And I guess it's probably somewhere, maybe both. I feel like if we just take the time and consider each case individually, maybe it'll have some more, it would determine whether we use a cocktail or sequential. So we've tested this in lab, of course, and cocktails have the advantage of more killing. And this, I'm sorry to blast you with so much data here. And this is a paper by Xian Mao, who was an undergrad in the lab and is now a resident in Massachusetts. And I apologize, this paper is not open access, but if you email me, I can get you a copy of it. Where basically we developed an algorithm to create a cocktail of phages based on lytic spectrum, and then we compared it with randomly chosen cocktails, and then each individual phage. And basically the greedy algorithm is the one on the left here. In terms of normalized growth, it was evaluating how little the bacteria grew in the presence of these cocktails. So then the next three plots are individuals, pairs of phages, and then the three phages alone, and then the control is just the bacteria. And at least in these cases, the greedy algorithm that we generated a cocktail with outperformed each of the phages alone. So you could potentially get more killing than an individual phage by combining them as a cocktail, and potentially less phage resistance. So OD600 on the right here is a plot of the bacterial density, and we just use it as it's basically optical density, so we use it as a proxy for bacterial growth. So when you have no phage, the bacteria population looks something like this over time. When you add a phage, it starts to grow and then crashes when the phage start replicating, and then you get resistance. This happens almost no matter what phage you use. Phage 2 is the same, or phage 3, you also get it. So each of these curves looks different, but you still get phage resistance. However, when you use a cocktail, you get a delayed time to resistance, and it's much less likely, especially in lab conditions. Also, cocktails have a broader spectrum. So this plot on the right is a host range. I realize I got to hurry here. So basically, what we do, the red squares, so CF1 through 11 are isolates, are bacteria strains, and PaO1 is a control lab strain. And then the phage along the top are the columns, and red means the phage cannot kill it, and green means the phage can. And so you'll see a difference in killing ability depending on the phage. But then when you combine them, obviously you get full killing, so it can increase the spectrum of your phage cocktail. Now, what about single sequential? So you get more killing over time, I would argue. Perhaps better for chronic infection management, as those you would see in CF. So again, you see the control with no phage. This is the OD600 plot again. So if you use the cocktail, it'll crash it, but then eventually, 48 hours later, it'll keep growing. You use the cocktail again, but it's evolved resistance, so it doesn't do anything. You try it again, and you get increased growth still. However, if you used just a phage, then you use a second phage and a third phage, you can manage infections this way and just keep killing the resistant mutants. Obviously, you have to have a well-designed sequence, and that's what we're studying to try and optimize that in lab. But it definitely can work, and we want to make sure we can come up with a nice regimen that will work in all cases. And that's part of the research we're doing, supported by CFRI. You also get more predictable resistance. And this was some older work where we sequenced resistant strains exposed to a single phage, a paraphage, or a cocktail. And the red is the percentage of mutations that are sort of off target, where the green is receptor associated mutations. So single phage, we were able to get predictable mutations in the receptors, and then cocktails, it was much less. We also got a lot more mutations when we used phage cocktails. So that's what we study at the Yale Center for Phage Biology and Therapy, where we do basic translational and clinical research, the whole bench to bedside, and environment to bench to bedside of drug development. And then we also go from the bedside back to the bench, and we analyze samples, and it's kind of the best place to work. I love it. And then this brings us to CF. So as you know, it predisposes individuals to infection from Pseudomonas, and I'm going to cut through this super quick. But I think CF is a great place to work in phage because there are some advantages to phage therapy over traditional antimicrobials. And then just to go over a quick case, in November 2017, we got inquiry to treat an AMR infection in a person with CF, and here's her strains antibiotic resistance. From the clinical lab, resistant to all these antibiotics. And here's her FEV1. Each one of these vertical lines is 1 week. And so she was a general decline for a few years. She'd go into hospital, give her, some IV meds, and then it would go up for a bit, and then it would return back to this trajectory. She'd go back to the hospital again for exacerbation. You can see the cluster of FEV1s is when she was in hospital. But then eventually she'd be in hospital, and then the antibiotics stopped giving her that bump. And so we treated this individual with phage for 7 days. And then, at the end of that phage therapy, which she inhaled via nebulizer, we had a big change in antibiotic sensitivity, and also in bacteria density. We also saw an improvement in FEV1. She gained 10% almost straight away. And then, after 2 courses, the strains were resensitized to multi-antibiotic classes. She had improved FEV1 for about 3 years. It was over 90% in 2020. And I just saw on Instagram that she's expecting, so congratulations. I'm not sure if you're watching, but excited. And she didn't have any hospitalizations to exacerbation for years. You can read about it here. I think there's a video also there featuring her and her family. Okay, so we also just are in the process of getting out a case series where I think it's just 9 cases. We've treated maybe 50 or so in our phage center, but we just did a report on 9 or so of them. That's like in revision, where we saw 3 log reduction in sputum density of bacteria, so 3 orders of magnitude. So it's like 99.9% reduction a couple weeks after phage therapy, and a 7.3% improvement in FEV1% predicted. The plot doesn't look as exciting as the number on the left, I realize. And we've also sequenced strains before, during, and after therapy. And so we have some microbiome analysis because I think individuals were worried that killing off Pseudomonas would allow some like Rothia or something to grow up uh, and outcompete the other stuff and cause maybe a worse problem than we started with. However, we don't see that based on these diversity metrics. It does not seem like it impacts that reducing Pseudomonas does not seem to open up a new niche for some of these other bugs. Obviously, this is just 9 cases, and this is more to be done. But I think more interesting to me is that the isolates collected before and after phage therapy show changes that we can predict in lab assays. So this plot here on the right is with pre and post isolates that we collected before phage therapy and after phage therapy, where we treated with a phage that uses the type 4 pilus as its receptor, which is, I think, one of the first examples I showed. Well, these are clinical isolates, right? So, and on the top here is airway epithelial, percent attached CFU. This is work performed by Gail Stanley. And then on the right is after phage therapy, we pulled Pseudomonas out of the sputum, and then we tested their attachment to airway epithelium, and then we saw a reduction which is comparable to that of a pilus knockout. On the bottom plot here, it's piocyanin production, which is a virulence factor that just causes damage to lungs. And we saw a similar result in the lab, and then also in the strains that we pulled from individuals treated with this phage that utilizes the type 4 pilus. And I apologize for speaking so quickly. I'm just trying to be sure that we catch up on the schedule. Also, when we looked at just LPS, we see the same thing. So some of these phages utilize the lipopolysaccharide of the cell wall as a receptor binding sites, and we see that the strains isolated after that are phage resistant have a reduced amount of cell wall, so it's thinner, I assume, and we also see a decreased production of extracellular elastase, which we see in the lab isolates when we force resistance. So we see a similar trade-off occurring from the individual. So in conclusion, to bring us up here is that I think phage can force predictable trade-offs in vitro for sure, based on some of this data and others, which has been supported by CFRI. Phage can be safely deployed via nebulizer to treat pulmonary infections associated with CF. We've never had a severe adverse event associated with phage, and so that's really great. And I haven't heard any even minor side effects except for some fatigue and maybe a slight fever shortly after. And I think that might be one of these Hersheimer reactions, but we're still looking into that. We can also see, I believe, clinically meaningful trade-offs that can be forced in vivo. At least the previous 2 slides seem to suggest that, and we're looking at this more. So I think given all that, changes in status, like an improved FEV1 could be attributed to phage-based intervention. There's obviously more work to be done, but at least in these one-off cases, and the number of those is really piling up, and everything looks like it's trending in the right direction. I think that phages might actually be doing something in a lot of these infections. We also have some non-responders that we really need to dig in and figure out what's going on. But that's part of research. Was figuring out what we're doing right and what we need to improve on, and see if we can do that, and figure out what's really happening here. And yeah, so that's our phage program. And I think, with that, I have to thank the CF community, because absolutely none of this would be possible without all of you, CFRI and others. Like, you guys have been the greatest, emotional support for research out there. and just your like optimism and kindness and willingness to share information, samples, everything has really made all this possible. And I couldn't ask for like a better partner at all, ever, than you guys. So thank you. Thank you. Also, Paul Turner, who directs the Phage Center, is great. John Koff's our medical director. Katie Courtwright is like this amazing postdoc who did tons of this work. And then we have a huge lab here, and visiting docs and ID specialists and research assistants and postdocs. And we always have people rotating through. And if you want to work with us, send me an email because we're always looking and I would love to work with you. So thank you for seeing through this, and I apologize for not being there in person. But again, you really don't want me there in person. The QR code on the left here is my contact details with my phone, so which is in silent. But you can text me if you want or email me, and then the only social I use is Instagram. But with that, I guess I could take questions if there's time, and I hope that I caught us up a little bit on the schedule.